All right, again, a very good morning to you all. My name is Senthil Sarvanamuthu, and I'm the founder and CEO for ONS Bio LLC, the distribution partner for ROKIT Healthcare here in the US Midwest, just like how I have Chris from the West Coast managing the West Coast territory through his company, Indicate Technologies. So first of all, I thank Dr. Ramaswamy Majumdar and Dr. Shankar for this opportunity to connect with you all today and also talk about the bioprinting as a powerful research tool and its potential as a therapeutic platform in the context of some of the emerging mega trends in science and healthcare. Just a little bit about myself. After my PhD from the Medical School Hanover in Germany, I spent 7+ years as a postdoctoral researcher in cell molecular biology at the NIH, Bethesda, Maryland. So during those times, whenever we had discussions with cancer researchers, two words always kept coming up, and that would be tumor microenvironment. So we talk a lot about that. I'm sure you all, as cancer researchers, also appreciate the same words. So if we can better map and visualize and manipulate, excuse me, the three-dimensional tumor microenvironment that dictates cancer behavior, then that would take us closer to evolving effective novel therapies for cancers and also improve existing ones. This idea is also one of the many important reasons that got me personally interested in bioprinting. There are various methods of 3D visualization today, but a 3D biomanufacturing technology that can compete with bioprinting, about which we'll be talking today, is unique. The 3D bioprinting is very unique in the sense that it allows one to build and map a tissue structure according to one's own research design. So the presentation today is kind of centered around the broader theme of engineering on-demand tissue systems using 4D bioprinting, and I'll get to what is 3D and or what is 4D bioprinting in a bit for research and clinic. And as I mentioned before, I realized your interest is mostly in cancer, so I took the liberty to change the title as Cancer World Isn't Flat, using bioprinting to approach cancer complexity in 4 dimensions in order to make it more focused and interesting and relevant for you. I will give a snapshot of why bioprinting is rising to attention in cancer research today as an application focus. But in general, it's important for me to say ahead of time that I want you all to think about bioprinting as a platform technology that can be applied for across all the major disciplines of science, not just cancer research. And so you can try and use some of these ideas that we'll discuss today in your own workflows and improve the quality of research by adding more personalization, automation, and on-demand fabrication to your research area. So with that, okay. I'm sure you can guess the image on this slide. You've probably even seen it many times. So this is a 3-dimensional simulation of tumor progression. In the left, you will see that each color that you see here represents a different mutation. The more successful one of these mutations is at reproducing, the more its color dominates the tumor on the right. On the figure in the right, what we are seeing are bulges of colors exiting from the core demonstrating how tumor cells or aggregates can outgrow, migrate, and squeeze into healthy tissues surrounding the tumor. So the truth of the matter here is whether we fight it or not, cancer is an extremely heterogeneous tissue made of different types of cancer cells. We need a better system to study and model both its architectural content and its dynamic nature. So we know that not only is cancer tissue heterogeneous in itself, it is also heterogeneous in its tumor microenvironment, which is what I was referring to earlier, the TME. So in general, the tumor microenvironment is known to be comprised of there's 5 cell types: the cancer cells, the cancer-associated fibroblasts, immune cells, endothelial cells, and cancer-specific ECM. And the traditional experiments in cell culture systems, which where one cell type is provided with abundant nutrients and oxygen, they have been used to do much of our experiments towards understanding of cancer metabolism. But we know that many differences that have been observed between the metabolism of tumors and the metabolism of cancer cells growing in monoculture. So there is always a lot of difference between the in vitro 2D models and the actual situation inside the body. Now, these differences are at least in part due to the presence of non-malignant cells in the tumor microenvironment and the resulting interactions between those cells and cancer cells. And there is also very little known about how the metabolism of various tumor stromal cell types differs from that of the cancer cells, and how this difference might inform therapeutic targeting of metabolic pathways. So there is a lot of unknowns here, and that's kind of what I'm getting at. The traditional cell culture systems are based on 2D culture or 3D spheroids of randomized cell aggregates. And the other popular ones are the cell line derived xenograft mouse models or patient xenograft mouse models. Now, regardless of which of these systems that we use, they all have intrinsic limitations. For example, and some of these are listed here, kind of summarized. But to touch upon a few, 2D mono cell culture on a flat polystyrene surface completely neglects both the mechanical and biochemical environment that you need to model cancer. 3D spheroids and organoids may have the added 3D built into them, but they often yield the problem of necrotic cores and very much restricted in their flexibility to control cell positioning and porosity variations. The CDX and PDX mouse models may have served as the best options or alternatives to mimic the in vivo complex tumor microenvironment. But as we all know, the human tumors growing in mice have intrinsic difficulties mimicking the human immune system interactions. So no wonder for one of this report that's shown here from the IQVIA Data Science, Human Data Science Institute, oncology studies have an extremely low success rate in getting a drug candidate passed through the 2D, 3D cell culture, animal studies, and human trials all the way to the market. Now, despite accounting for almost 30% of the R&D product pipeline across the board, the success rate for the oncology studies actually is only 8%. And that's a very inefficient outcome. to say the least. So this is kind of the context and exactly the reason why the technology of bioprinting is now rising to the attention of cancer researchers worldwide. Bioprinting, for those of you who haven't heard about bioprinting yet, it is a perfect combination of computer science, biology, instrumentation, engineering, and material science. What it does is it brings the benefits of personalization, autologous solutions, and fine details to human health applications. In every sense, 3D bioprinting is an additive manufacturing process in which cells, biopolymers, and growth factors can be combined to create tissue-like structures that imitate native human tissues. Because these printed living tissues are often further incubated to have them proliferate, mature, and change over time, so clearly there is a dimension of time here that makes it appropriate to call 4D printing or 4D bioprinting instead of 3D, because we're talking about living entities that change over time. So when you take into account the temporal component, then it makes sense to call it as 4D bioprinting, and that's what you would see in most of the discussions that will follow. But for simplicity, we may switch back and forth and say 3D bioprinting, 4D bioprinting. So I hope that doesn't confuse. All right. So according to a computer-aided design that one comes up with, bioprinting unleashes the potential to closely recapitulate the complex compositions of biomaterials, cell types, and vascular networks that cancer can exist in. So to give a broad summary of what are the different components of bioprinting technology, we can group them into three. And those are, one, the biomaterials themselves. And then number two is the bioprinter hardware, the actual equipment that prints. And then equally important is the design or the software component, where you try and digitalize the information that you have from an in vivo tissue or organ or anything that you fabricate for your own experiment. And that kind of generates the code that's needed for the hardware to go use the materials that supply and recreate. So these are some this is a clip of bioprinting in action. Now, early 3D bioprinting technologies may have been limited to rigid materials, typically made from hard plastics, but things have evolved and the field is growing rapidly. Now, 4D bioprinting, as we know, offers fabrication with soft fluidic gels, and we have ways to further polymerize by use of light, temperature, and chemical reactions to cross-link the biomaterial. So even cellular structures can be printed and preserve the architectural integrity until further incubation or during the incubation as you do the experiments. So you might now be imagining why and how bioprinting is actually used in cancer research. Well, this slide is a good intro example to demonstrate that potential. In this, what you see is a bioprinting has been successfully used in this example to model glioblastoma tumor microenvironment. In a Nature article by Ozpolot Lab at Penn State, the group demonstrated printing multiple cell types with a gas-permeable channel that closely mimics the multi-layer brain tumor tissue with different oxygen gradients. To help you visualize, the ROKIT Healthcare's R&D team has printed this model in-house, and this clip will show you how things are layered to recreate a similar model. So as you can see here, you can print multiple layers in a single construct. And not only that, you can also print in any orientation that you deem fit for your experiment. In this case, what you're seeing is a horizontal orientation or layering of those different layers. You can also flip and then do it in a vertical orientation. This is just to drive home the point that it is very flexible to do and orient as you deem fit for your own experiment. So bioprinting has been applied to create metastatic models as well. In this slide, what you're seeing is the research by Panos Mortari group at the University of Minnesota. What they have done is the group has created a 3D printed in vitro tumor model integrating tumor cells, endothelial cell-lined vascular conduits, and included biochemical signals within a fibroblast-laden fibrin gel. So the tumor cell invasion of the surrounding matrix and into the vasculature are mediated by the EGF and VEGF gradients, which are themselves dynamically generated by the 3D printed stimuli-responsive microcapsules that you see here around those channels. Now, the stimulus in this case is a laser. So when you shine the laser, then that leads to the release of these EGF and VEGF, which forms a gradient and so on. For simplicity and also to keep time, I'm not going to go into the fine nitty-gritties, and this presentation will be provided to you later on. And each of these slides has the specific publication references, and you can always revisit that. And if we have more time, we can talk about these during our discussion. But the idea is to show some of the examples that kind of demonstrate the power and potential of the technology. So far, the point I'm trying to drive home is that bioprinting brings novel key values that overcome some of the age-old problems that traditional culture systems have always had. And by no means is bioprinting the silver bullet. It's a perfect technology. It's a technology that is moving towards some level of perfection, but neither can it completely replace all the traditional techniques, nor can it be the single solution for all the inadequacies with any other existing methods or techniques. But what it really does is that it has immense potential to translate the existing techniques to the next level of complexity and functionality because of the very unique value additions that it can give to your current workflow. And to summarize these, there are three aspects in which it can give you a lot of value. One is in your ability to create architecturally complex models. For example, in one of the bioprinted cancer models, a gradient of hydrogel matrix was also developed in 3D to facilitate directional cell migration via controlled deposition of bioink. And that kind of mimics the physiochemical environment of cancer cells. Now, not only that, you can also control stiffness, spatial distribution of biochemical factors, and also you can do free-form design. There is one particular technique which you can see here in this short clip, which is called the FRESH technique, which is free-form reversible embedding of suspended hydrogel. That's the expansion for FRESH. It's one of the recent techniques of extrusion-based bioprinting methods for mimicking complex native architecture of biological tissues. Now, using this technique, there are examples of neuroblastoma models that have been developed with sodium alginate and so on. But the point is, bioprinting provides you greater control in terms of free-form design and your ability to do architecturally complex tumor modeling. The second is, as you can also see these things in action, is the ability to do automated dispensing. And that what that means is it provides the ability to do reproducible models. The experimental repeatability is very important, and that can be achieved here. And also, as you can see here, high throughput is possible, and you can do all kinds of cross-linking functions as you try out different materials to fabricate. So those are some advantages. And then lastly, but still important, is the fact that we can also use bioprinting to explore diverse biomaterials, ranging from bioplastics, going all the way to hydrogels to cellular solutions. So I'm not just talking about just synthetic or naturally derived biomaterials from animals or wood or ocean algae or all these different sources of biomaterials that are out there. I am actually also talking about something more important, which is the autologous liquids or solutions from patients' own material that can be harvested, such as human plasma or patient-derived tumor primary cells or stromal vascular fractions from the adipose tissue. Now, those can also be used as biomaterials to fabricate all these different structures. So when you put all these values together with the ability to use cell sources from patient biopsies in the printer, you've got a translatable technology that can be advanced to the clinic and also used for high throughput applications for more effective regenerative medicine strategies or evolving more robust therapy screening methods. And what you're seeing here, I may or may not have time later on, is a snapshot of the interior of ROKIT Healthcare's newest lineup of pretty advanced bioprinter, which is the Dr. INVIVO 4D6 that comes with multiple rotors on a rotating turret that gives you the ability to use multiple materials at the same time, plus the ability to do things in high throughput because of its high throughput liquid handling ability. So to suit any format, be it a 96 well or a 384 well, and also the ability to handle variety of biomaterials using different print heads, as you can see here, the hot melt or the extruder module, or the syringe dispensers for soft hydrogels. We'll get to discuss these in more detail later on, but so this is something that you may want to get acquainted with if you're totally new to bioprinting. All right. So more importantly than what we have discussed now, any ideal model of cancer tissue or human tissue would need to account for the environment that they are exposed to during and after the process of biofabrication. So one needs to carefully control the environment in which you place the cells from start to finish, and also during incubation, so that the printed product remains as closely matched in its properties to what you would find in vivo. So only then can it serve as a true representative model that can be used in your experiments and gather meaningful data. So in the case of mammalian and human cells, cell viability, proliferation function are critically dependent on, we know, the environmental cues such as temperature, humidity, pH, and sterility. So the INVIVO 46 and its cousin, which is 42, about which also I'll be talking about later. Now, these bioprinters are pretty advanced in their ability to have, in the fact that they come with, at least the 46 comes with a built-in cell incubator or a bioreactor function that's able to maintain a neutral pH and a 95% humid and a very sterile environment, offering precise temperature control that would optimize the success of human tissue fabrication and research. And there is a lot of emphasis on the bioprinter's ability to control the environment during printing to keep the cells happy. And that is kind of the direction in which many of the bioprinting development is moving towards. And INVIVO 46 is kind of a trendsetter in this regard and is leading the way. All right, ROKIT Healthcare as a company is a pioneer in bioprinting field, and it's not just a manufacturer of bioprinters, it also has dedicated R&D, and it's a well-integrated regenerative medicine company, so to speak. So ROKIT Healthcare as a bioprinting community is 250 labs strong and is extended over 25 countries. And these users range from academia, industry to hospitals, and they also span across various disciplines of research and departments. So in this half of the session, my goal is to take you through some real life examples of bioprinting in cancer research and therapy development. These are taken directly from ROKIT Healthcare's Dr. INVIVO bioprinter user publications. And they are very good demonstrations of why 3D bioprinting is being used by cancer researchers to better mimic tumor biology and predict cancer responses to drugs. So let's go here. The first journal publication comes from a group that created a 3D cell culture printing model for bladder cancer. The bioink used was created with a mix of bladder cancer cell line and gelatin methacrylate, which is a form of gelatin that can be cross-linked by exposure to UV light at 365 nanometers. Now, branched construct models of, say, 1 by 1 by 0.01 millimeter size were created with two cell types, one using cell line 5637 and the other one using T24 cell line. Now, each of these models was then compared, the 3D versus 2D, for cell proliferation, cell-to-cell interaction, and how we assess cell-to-cell interaction is through the by looking at E-cadherin and N-cadherin proteins, and also cell survivability against common bladder cancer drugs such as rapamycin and BCG. So the first thing that the research group validated was that the 3D bioprinted construct after UV cross-linking could maintain structural integrity over the lifetime of the printer, of the printed structure at the cross-linking times of 120 seconds and 0.08 millimeter height in layer structure. The group found that the structure of 3D bladder cancer cells could maintain structure after incubation for 5 days. So the structural integrity is very well preserved. Now, one major finding from the comparison of 3D printed bladder cancer cells to the 2D cell culture model was the difference in the proliferation rate. As you can see here, over a 3-day period, regardless of the cancer cell line, whether it was the 5637 or the T24, the bladder cancer cells showed higher growth rates in the printed 3D cell culture model than in the 2D cell culture. And this difference became apparent starting on the second day of the culturing. And here, another difference is highlighted. The difference seen here is when the bladder cancer cell lines were treated with rapamycin, which, as we know, is known to induce autophagy of cancer cells by inhibiting mTOR and its mediated signaling pathways involving the P70S6K and the 4EBP1. So the message here is that compared to the 2D, the 3D cell culture environment showed more residual levels of expression of mTOR and P70 and the 4E proteins, which shows that the drugs had less effect on killing the cancer cells in 3D cell culture model than in the 2D model. Finally, we have the 2D and 3D bladder cancer cell models when compared in terms of N-cadherin proteins, which we know are basically proteins upregulated in epithelial cells that lose these cell-to-cell interactions and become more mobile and invasive in behavior. There was a significant difference in the amount of N-cadherin in the 3D cell culture model as opposed to the 2D when TGF-beta1, which is a stimulant of EMT, the epithelial mesenchymal transition, was applied to the cancer cells. So the group concludes that the bladder cancer cells in 3D exhibit higher cell proliferation, higher drug resistance, and stronger cell-to-cell interactions overall than the 2D models. And because of this opposite character from the 2D cultured cancer cells, one can understand why sometimes up to 50% of the patients undergoing common bladder cancer drugs simply do not respond within the first 5 years of treatment. So the 3D model is essential for predicting and evaluating drug responses in the future and for developing accurate personalized medicines. Now, taking a step further from the previous publication that I discussed now, this second study by Han et al. Um, shows how to add compositional and dynamic complexity to a 3D bioprinted cancer tissue by creating extracellular matrix, microenvironment, and vasculature systems. So here, what the group created is an in vitro vascularized tumor microenvironment with controllable tumor spheroid sizes. First, the group printed 10 by 10 by zero point 0.6 millimeter sized extracellular matrix patches using alginate, fibrinogen, and gelatin composites mixed with lung fibroblasts and HUVEC endothelial cells. After double cross-linking of the alginate and fibrinogen with calcium chloride and thrombin, the bioprinted tissue structure was incubated to develop vascular networks, and upon this vascularized tissue, the glioblastoma spheroids of controlled sizes were seeded and further incubated to result in a kind of a vascularized tumor model. So they combined both. And as you can see, in the initial stages of cross-linking, the group validated that the tissue could first stay with structural integrity well into 7 days, as you can see here, of incubation in the media. The lung fibroblasts and HUVEC cells printed had about 80 to 90% viability after printing. Because so two things that always come to mind: what about viability and also structural integrity? Now both of them checked okay. So this bioprinted tissue was also assessed for functional markers for microvessel formation. And the group noted that they could see high expression of actin and CD31, which are both markers for well-developed capillary networks. And this was further confirmed by measuring the area and thickness of blood vessels in the layers, indicating those on the day 7 were actually thicker and larger than on the day 4. Even the lumens, which are indicators of a mature blood vessel, could be observed here, and even without preformed blood vessel channels. Now, this method here shows there is an advantage of being able to produce large, open, vascularized tissue very easily and rapidly compared to other biofabrication methods. And that's a clear advantage using the bioprinting technique. So the major finding from this study, to summarize, is that there is a significant difference between the 3D tumor constructs with and without vascularized tissues. And the figure here shows that the vascularized tumor spheroids exhibited much more active proliferation than the non-vascularized ones. And that is evident in the size and morphology of the aggregates over 4 days. Both observed qualitatively as well as quantitatively in terms of area and volume. Furthermore, a look at the gene expression at mRNA level of encaturin and vimentin, which are involved in angiogenesis and invasion of cancer cells, was several times higher in vascularized tumors than in non-vascularized ones. Last but not the least, the group also treated these vascularized tumors with anti-cancer drug, which is TMZ. I believe that is temozolomide, and the angiogenic inhibitor sunitinib, which is shown here as SU. What they conclude is that the combined treatment involving these two drugs, TMZ and SU, was more effective than the TMZ alone for these multicellular tumor spheroids that were surrounded by blood vessels, which indicates the feasibility of the tumor microenvironment for in vitro testing of drug efficacy. Now, these results suggest that the bioprinted vascularized tumor is highly useful for understanding tumor biology, as well as for in vitro drug testing. All right, so this bioprinted model here is another study that takes the existing co-culture model to the whole new level. For example, while the co-culture model may simply provide cell-to-cell interactions, using the bioprinting example, it's possible to offer the presence of mature vascularized system and examine the interaction of that with tumor cells in not in a system to system interaction level, not just at a cell to cell interaction level. So that brings in some value to the study. Now, stepping up the low complexity to higher complexity models, this is a third study that shows how high throughput bioprinting and patient-derived tumor organoids can be combined to create a drug screening platform. Here, tumor organoids are combined with patient-derived cancer-associated fibroblasts and endothelial cells to create what is called the assembloids. These assembloids are further printed in consistently controlled 10 microliter droplets in 96 well formats. So far, what we have discussed in detail is the technology of bioprinting and the reasons it is rising to attention in cancer research and some of the unique value proposition it offers to advance our understanding of tumor microenvironments. Now, the nice thing about this technology is that the cancers is just one of the many possible applications. As I mentioned before, this is a platform technology that can be applied across many disciplines of science and can employ various types of cells, biomaterials, and the 3D designs that are simply limited by only your imagination and your need, and your experimental need. So what you can realize is that the applications of bioprinting are numerous, and they range from tissue engineering to drug discovery to non-animal testing, either to complement existing animal models or eventually to completely replace them once the technology is perfected. So many different applications are possible. And here you see an example of some. And for today's talk, since we are already cutting short on time, it is not possible to go over everything in detail, but I'll give you a quick snapshot of some of the other possible applications, just touch upon them to give you a flavor of what's all possible, what's all out there. And this slide is very useful to get started with, because in this slide, what you're seeing is demonstration of how bioprinting is a platform technology. And what you can see here in this table is on the left in blue, hyperlinked numbers. And these are all publications that have been gathered in the kind of review articles from the last decade, demonstrating evidence about the ideal choice and combination of cells and biomaterials for various tissue-specific applications. And as you can see here, it covers a lot of important tissue types: cardiovascular, musculoskeletal, neural, hepatic, adipose, and skin tissue. And by no means this is an exhaustive list. And also, we know that the the global 3D bioprinting publication output is continuously growing at a rapid pace, clearly showing that bioprinting is going mainstream, and now might be a good time to get on board with this new technology. So in the last 2 years, the very first bioprinted, and this is an interesting point to note as well, that the bioprinted disease models are also are making their foray or entry into the clinical trial world. So in the last 2 years, the very first bioprinted disease models got into the clinical trials. As you can see here, some examples. And here is an example of the application of bioprinting for cardiovascular research in the heart patch regeneration. And I won't go into the fine details here, but it might be useful for you to see that the bioprinted heart shown in this study, in this example, has been beating for 6 months. And so the cells are pretty viable, and these kind of help one appreciate the power and potential for bioprinting to serve as a wonderful model system generator for studying some of the complex disease problems. And also these heart patches are designed to serve as cardiac regeneration graft patches for therapeutic purposes to treat chronic ischemic heart disease. And as an extension of this, it's also possible for one to imagine that since the bioprinting offers ability to finely control the architecture, one can create in vitro models of human pathological cardiac tissue by altering the various fine architectures and orientations, and that way come up with various different infill patterns that will mimic the behavior and microenvironment of different stages of pathological cardiac tissues, which will be useful to address different stages of progression in ischemic heart disease and so on. This is an example of the nerve regeneration, application in nerve regeneration. I don't want to go into the details of this, but one thing to note here is that the existing autogenous nerve graft is for treatment of nerve defects is a less than perfect technique because of the significant morbidity of the donor site. Now, in this example shown here, the group has used elastic nerve guidance conduits using nanoporous structures with materials like PLCL, and combined it with oriented 3D printed collagen hydrogels. And the resulting aligned collagen hydrogels enabled by 3D printing patterns appear to provide a preferable environment for nerve regeneration, providing a guidance path along the longitudinal direction. So this method may be further enhanced by incorporating various types of growth factors or bioactive molecules in the collagen, and kind of help do further research in nerve regeneration. This is a quick sample of a case where you don't have to always use living cells, and that's a question that many beginners ask, if bioprinting should always involve cells. In some cases, maybe it's not needed, depending on your experimental approach and goal. So here is an example where where one has used non-living materials And this is an example of development of a QR-coded drug delivery device that has been used to provide an optimized um, uh, drug delivery solution. And what the group intends to do here is to use printed oro-dispersible films like these to address problems of wrongful prescriptions to patients or overdose, and also to improve the solubility of drugs, in which is a significant challenge in the drug discovery field. So there are also these other interesting applications possible. So bioprinting is also being used in development of soft food and cell culture, and also to generate meat production, that 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 can take away the need for animal-based food production. So there are many other innovative, interesting areas of application possible. But what would be more exciting than the actual use of bioprinter for therapeutic application in the point of care scenario, or use of bioprinter for personalized autologous therapies. So what you're seeing here is the image of the ROKIT bioprinter in action in the operation theater. This is a photograph from a scene in diabetic foot ulcer clinic in India. India being one of the hardest hit countries for diabetic foot ulcer. What is being done here is the bioprinter being used to generate a breakthrough method of regenerative wound healing for chronic wounds for treating diabetic foot ulcer. And what you see here is the picture and graph of 40 patients tested in India and Korea, where 100% of the patients demonstrated reepithelialization of wounds in 4 weeks on an average. And these are all chronic wounds of patients who have had no improvement from traditional wound dressing materials and negative pressure wound therapies, which are common ones. So the wounds that remained open for months to years were able to close within 4 weeks using the bioprinted therapeutic approach that was developed by ROKIT Healthcare using the Dr. INVIVO bioprinter. So this is to highlight that the bioprinted patches using patients' autologous solutions in an on-demand way in the operating theater is happening right now to address real needs and problems in the hospital. There are more global clinical studies underway in US, Turkey, and Italy in the upcoming year. And we envision that this therapy can be applied to more skin conditions, ranging from burns to scars to bed sores and so on. Just remember how I mentioned about use of bioprinting as a combination of digital world and the bio world, how to bring them close. Well, if we move away from the hardware and biomaterials for a bit and shift our focus on advancements onto the digital software side of bioprinting, one can imagine that artificial intelligence informing the production of patient customized patch designs in a fast, reproducible way. And at ROKIT, they are already making progress towards that goal. And this video here will give a quick view of that effort. In the interest of time, I will quickly scroll through this video. So what's happening here is basically the patient's wound is scanned by the scanner, and then the data is digitized, and then the 3D files are generated that are custom made for the custom made patches for specific wounds, and followed by that the data is processed, and then the ROKIT bioprinter starts printing those patches using a combination of bioinks and patient-derived material from the patient's adipocyte derived SPF. And those patches are very custom made, and so they fit right into the wound area, and that helps in generation, in regeneration of that area, and a complete healing. So in the context of all these things that we have discussed so far, ROKIT Healthcare as a company provides tissue engineering platform, which is the bioprinters, Dr. INVIVO 42 and 46. And as I mentioned before, ROKIT is also a well-rounded bioprinting company and regenerative medicine company. So they have other materials as well, the biomaterials that are an integral component and an important component to giving a well-rounded solution. So they do have those capabilities, and above all, it's important for me to also point out that ROKIT as an organization is very keen on providing the online and offline curriculum-based training and education, towards its goal of popularizing bioprinting. So they do have the Organ Regeneration Academy that provides the curriculum-based training and education for all kinds of users, whether it's beginners or advanced. And it also serves as a forum and a platform for online and offline collaborative R&D and business development. This is to give you a quick idea of the Dr. INVIVO bioprinting platforms offered by ROKIT. This is the Dr. INVIVO 4D or 42, which when it was launched was the world's first desktop bioprinter to have a germicidal UV and HEPA cleaning chamber biosafety cabinet incorporated into the bioprinter as a standalone. And it is also the only desktop bioprinter to have been used in a clinical human study, the one that I mentioned about use in diabetic foot ulcer a short while ago. We have, as I mentioned before, 250 plus reference labs established worldwide, 30 plus publications and international recognitions already on various fronts. The printer can handle wide range of materials, bioinks, which could be soft hydrogels with or without cells, powder materials like hydroxyapatite, chitosan, so on and so forth, and synthetic polymers as filaments, PCL, PLGA, PLLA. These are just examples, but you can throw in any mix of materials that you may be customizing for your own research, pellets, powders. So it has the capability to do all of these because of the advanced temperature and pressure ranges of its print heads and the various other features that come with these equipment. And one important thing to keep in mind is as ROKIT embarks on taking the technology from benchside to bedside, It's important to note that the human body is not a flat layer by layer tissue in every case. It is a very complex system. So much of what we see here are curved, and to recreate those level of curved architecture, it involves it requires the printer to have the ability to do curve printing in z-axis, and also it is important to, for any bioprinting solution to move into the um, therapeutic arena. One needs to carefully control the process of sterilization, and also how the cells are incubated during and post printing. So thinking about all of these, plus also gathering feedback from existing users, ROKIT has improvised the existing INVIVO platform to come up with a more robust medical product development ready system, which is the Dr. INVIVO 46. And in the normal times, we would have had a lot of in-person opportunity to do in-person demo for these equipment, but since the onset of COVID, everything has changed. So I'll quickly give you a 20-second video of the INVIVO 46 in action. But we can always revisit this and discuss more whenever you're ready to talk about this. Lastly, I want to also touch upon the fact that the ROKIT's proprietary human cell-derived whole extracellular ECM called the humatine is a ideal coating material comprising of 300 proteins and associated cytokines, because it harvests the extracellular matrix in a native form. And that is very important, because most of the existing bioprinting material, or most commonly used ones, are single component. It could be collagen, or it could be fibrinogen, whatever. That doesn't really do justice to recapitulate the extracellular matrix that you actually find in the cells, because we know it is a gamut of 300 proteins and growth factors. So trying to replace 300 by just one doesn't do justice to the process of recreating or mimicking the human structure. So the humatine is a wonderful biomaterial that can come in handy. I will skip these portions, but these are extensions of ROKIT's capabilities on the biomaterial and extracellular, human extracellular matrix components available to go in alongside the bioprinter. So I will say that the whether you are a beginner to bioprinting, or whether you are an advanced user, you would probably be always looking for updates and ability to share and gain knowledge from people in the same field, and adopting the bioprinting technology and putting it to the best use for your own research efforts. So to facilitate that, as an industry pioneer, ROKIT Healthcare is also dedicated to creating a hub or a community of users to bring the power of combined knowledge and expertise through its e-learning platform for all things bioprinting. And so that's something that you also become part of as you adopt the ROKIT's technology. So as a takeaway, there are many things that you can talk about bioprinting and its usefulness for your research. But to simply summarize, the value proposition falls into these three categories. One is it reduces the overall time and cost of 3D tumor modeling process. It increases the accuracy of the developed model to facilitate a better drug screening assay without much dependence on animal models, or it may be, or the data helps support whatever you may be gathering from animal models. It can work both ways. And also it broadens the project scope, and it also brings in the power of automation to your workflow. So all of these things are reasons for you to think about as you consider adopting this technology. And more importantly, we all know that in the current day and age, it's an era of precision and regenerative medicine. And the era of blockbuster cookie cutter drugs is coming to an end. So things have to be more personalized, because we appreciate the fine differences between individuals' genetic makeup and the need for personalization for the drugs to be more effective. So in this new era, bioprinting is not just a revolution that's underway for the way we create and use model systems for answering basic biological questions. It is actually revolutionary in the way that you practice medicine. Instead of using systems that rely on treatments of only symptoms using small molecules or monotherapies, bioprinting, what it does is it shifts the focus towards harnessing body's own healing potential hidden in the stem cells. And the future of medicine is already being realized today by these pioneering technologies in various disciplines of science. And I'm really glad that your group is also at least interested in this new promising technology. And I hope you will consider adopting this into your workflow for cancer research. I guess that's all I have for today. I really appreciate your patience. My apologies if I took a little longer.
Oh, it was great presentation. Yeah, I am going to be a little bit in think for time because I have another meeting. But it was a great introduction. What an amazing technology, I think, to kind of be more closer to human conditions as we, either try to understand mechanisms of disease or try to understand the novel drug therapies on how it works or how it doesn't work, and then fabulously, of course, recreating human tissue that you can actually use to improve healing or other conditions. So fabulous, kind of work that you're all doing. I think we certainly have, particularly what we are working on in certain specific type of breast cancers that have very, very slow, growth, as well as difficult to grow in vitro studies. One of the difficulties we have with this lobular cancer, breast cancer cell lines, is to do grow it in the 2D cultures that we normally use, because it's just extraordinarily slow growing. And then with that, then to try different treatments are all difficult. Certainly, particularly just talking about what we are doing in that area, I think it'll be very interesting to think about these options to understand this cancer better, as well as to try with novel therapies. But, as far as taking it, getting this technology to us, I do think we should involve the rest of the breast cancer research group. The basic science group is far better than the clinical group to listen to this. And we do have an OSU Comprehensive Cancer Center breast research symposium or meeting that we do every month. So what I would suggest, I would say, is that I discuss this with that group, that whole translational group, and see whether this is a discussion that we could have again in one of those meetings where you could present a short presentation, or I could share some of these slides, and then see how best this is a tool that can be used with multiple by multiple teams. And I know I'm still kind of stuck with breast cancer. There's, of course, the whole cancer group.
Right.
But we could start that as probably what I think would be the next steps. So, but I'll let, I'm sure Sharmila and Shankar Ishwar has some questions.
Oh, for me, I think it is like a very new introduction. I read a little bit about it, but it is absolutely new to me. And I think reading the references that you have cited in your presentation will be important for me to figure out how we can use this technology in the current research that we are doing. Definitely, as Dr. Ramaswamy has been telling, that these ILC cancers, the lobular cancer, are very slow growing, particularly when we take it to the mouse model. It takes 8 to 10 months to grow a tumor. So if that can be cut short to 3 days using this bioprinter, that will be, fabulous, I would say. Like, that's the time is the thing that it is stopping us from getting into the mouse model. But, I don't know how the slow growing cells will behave in these 3D printed models, but we certainly can try that. And again, group setting or in interest of multiple PIs will be important. But I definitely am excited to see how we can cut short that 10 months into 3 days and do some drug testing. I mean, that's going to be really great. And one key question that I have is like, how do you read out? Like, you use your drug, like a 2D culture in a 96-well plate, we use different assay systems. But for a 3D printed organoid or the material, how do you read out these? Are they same as a 2D culture?
Yes, for most part, it is the same. Wherever for example, you use 2D cell culture, what are the normal readouts that you use? You probably do microscopic phenotypic analysis or gene expression, or what other assays do you use?
We do the pharmazone, like the MTT assay for regular basis. Like, you throw in some drugs and you look at the effect of the drug by doing MTT assay, more like a mitochondrial activity assay. But is that possible to do with this?
Yes.
Or there are other?
I'm not the expert in that. You guys know probably way more than I do. Dae, do you know of any other assays that have been used? But some of these publications are going to give you the answer to all these questions, especially a couple of reviews. I'll send them as well.
Yeah, I think that will be something that we need to look into to see like how we can, do the readout is an important thing to
the
consider.
Drug testing part, what is the readout? Is proliferation? Are we looking at that? Cell death, in which states? Well, how do you do that in your 3D models?
Yeah. So to chime with what Centil said, more or less, the readouts or the analytical portions of your experimentations are not really going to change with the 3D bioprinted models. Because at the end of the day, when you have printed your models with the bioprinter, what you're basically creating is your cellular tissues that are no longer standing in 2D, but in 3D with the added hydrogels that you have put into the bioinks, right? But as they are cultured in the incubators, those hydrogels you can kind of think of them as those hydrogels, which are the engineered ECM components. They will be replaced by the building blocks of the actual ECM that the cells begin to produce in three dimensions. But anyways, what you will get out is basically three-dimensional cellular, three-dimensional tissues that you can either section in order to analyze, or you can just apply your traditional MTT test and then read out how many cells basically die off at the end of that test. Basically, whatever you do with the tissues will not necessarily change. You can just think of these products as something that has greater volume and perhaps greater proliferation rates from 2D, but the analytical portion, the actual activities that you do with these tissues will not change from what you do with two-dimensional cell culture results or, isolated tissues from the mice, for example. Yeah.
So the other key question that I have, like, if we think about in the future, are you going to are we be able to consult you guys in designing our experiment? Like,
Yeah,
what is technical help?
Mm-hmm.
Yes, we realize that this is a new technology, and so you're going to need a lot of support until you get comfortable. So, what comes when you get the technology is unlimited support over phone, email, and Skype, and all the online tools for more than a year. In some cases, even 2 years, by which time the goal for us is to get you up to speed on the technology and be very comfortable in applying this. And then it's all left to your imagination. And you can easily take it from there. But in terms of designing the design files, the software component or the hardware, no worries. We are here to support. But at this point, then I'd also like to bring up this before it's too late for Dr. Ramaswamy to leave. ROKIT is very supportive of all these efforts. And so we would like to also mention here that if you have ideas where you may be planning a grant based on this, bringing this technology in, and if you need any support, letter of support, or any collaborative research possibilities exist at any level, maybe Dai, you should tell more about that. ROKIT is very open for that, and please do take advantage of that.
Great.
That's really good to know, actually.
Yeah.
That's really good to know that, because, if we can have, obviously that's the biggest thing, to get grants to support this work.
If I may say, I think my CD38 grant had a small part in this specific aim, including the 3D bioprinting. That's how I got introduced to it, in order to see the immune signatures that in a 3D scenario infiltrated into the triple negative breast cancer. And in fact, they gave me a letter of support, and they also helped me design that experiment. Yeah. When I pass on that proposal to you, it's a part of the specific aim that was introduced.
And the big advantage with ROKIT is it's more like there are many companies that manufacture the equipment and then don't do much in terms of applying it in the real world. But as I've been telling again and again, ROKIT is more a well-rounded regenerative medicine company that not only talks the talk but also does the walk. So they basically have a lot of in-house R&D projects going on for various applications. So all that knowledge and experience also gets transferred to its user base. So, and that's something important, because equipment is one thing, but the know-how is critical. And ROKIT is very generous to share that, and they want that to happen and see more of, because there is no way we can perfect this technology by just working alone. It has to be done together. And the academic partners are a key player in that. Dai, are you there?
Yeah.
Yeah.
And
So would you like to add
at the
something?
same time, we believe that bioprinting has a very unique potential to add values to some of the traditional medical therapies that have yet to solve age-old problems. And so other than the basic science work, we are also very concerned about translations of the bioprinting technology into the operating room and the surgical rooms. And some of the works that we are doing in communication with agencies like the FDA or the European Medicines Agency are all geared towards providing at least a little bit of guidance for those researchers who have the same mission for translating what they do on the bench to the operating room using this relatively novel technology, but powerful technology called bioprinting. So on those fronts, we could also, and we look forward to working with our users and partners to develop, novel ways that bioprinting can be clinically translated. And by the way, in response to Sir Miller, your question about the analyzing 3D bioprinted models, I also just very briefly looked into some of the abstracts for publications that have been out. And one thing I forgot to mention is that with the 3D bioprinted tissues, if the MTT, assays like the MTT, if there's any trouble with the reagents basically penetrating into the 3D models because of the volumes and whatsoever, there are many ways. There are actually kits that also allow you to basically dissociate the 3D tissues into models that can be, I guess, more available for penetration by the reagents. And there are kits specific for 3D cell culture, and these can be applied just as readily for 3D bioprinted tissues. So I just wanted to add that.
Sure. Thank you so much.
Yeah.
Great.
So,
Thank you so much. And I mean, I can step out, and if others want to stay, that's fine. But what I think, it'd be great. You said you would send this, presentation to us, and that'll help us to look at those publications. And then I definitely, very, very enthusiastic, obviously, and the next steps, right, in what we do and how we impact research as well as patient care. But I will also take this then to our larger group and see what the interest levels are, and maybe have another presentation which involves more of the basic researchers who would, I think, be interested in this, and then we can go to the next steps after that.
Sure.
So.
Sure, absolutely, yes, and I'll send the publications as well and the link to this. This is the last thing I want to say before you leave. We strongly believe, and many others in the field also believe, that the bioprinting applications are going to become such an integral part of doing science that most journals will, in the next few years, start asking people for this data from the printed tissues to go as an in-between the animal or 2D cell culture and the clinical, the human tissues. And that is just unavoidable. It's happening. So the sooner we get in, we have a chance to make some high impact before it becomes the norm.
Absolutely. No, I hear you. And this has happened before. We were, that's with organoid and PDX, and now we have this. So I don't doubt that.
Or with the NGS. There was a time when NGS, it's not for me, it's not for them. But now, who doesn't use that for any gene expression data?
Sure, absolutely. Yes, everything. Yeah, you're absolutely right. So no, but it's a fascinating technology. So we'll be in touch for sure. And I really appreciate the time and the details of that was very helpful because it's very new to us. So it's excellent. So thank you. Thank you so much.
Thank you for your time and interest, really. I appreciate this opportunity.
Thanks, Ishwar, for bringing this to us. So we really appreciate that.
When we had a conversation, I thought this would be a better model for all of us. And as I mentioned before, I had put this in the new proposal, but we can always discuss this. As I bring the presentation to you, we can always discuss it.
Absolutely. Fabulous. We'll be
and I'm based here in Cleveland, and I'm responsible for your area, so you have me all the time.
Great. Wonderful. Great resource. Thank you so much. Thanks to you all for taking the time.
Thank you.
Appreciate it.
Thank you, everybody. Thanks.
Thank you.
Bye-bye.